Assalamu alaikum. So in this lecture, we'll be talking about genetic disorders and modes of inheritance. And this lecture is divided into two lectures, actually. The first one will be talking about Mendelian genetic disorders. So genetic disorders can be classified into two types, either monogenic or multigenic. Monogenic disorders are caused by a single gene, and that's why they're known as Mendelian genetic disorders. Usually they are confined to a single organ, except if the protein is common in different organs, like enzymes, for example, then multiple organs will be affected. Now, multigenic disorders are caused by mutations of several genes. Now, if the environment plays a role, then we call them multifactorial. Okay, if environment or if pathogens do affect the disorder, then they're called multifactorial. And this is the topic for next lecture. Now, these disorders usually they are complex. They are also unpredictable. Now, so far they are unpredictable. So let's talk about Mendelian genetic disorders or disorders. So now these again they are caused by single genes. They are mainly autosomal, meaning that you would have a defect mutation in a gene located in chromosome 1 to 22. Now you also have X-linked disorders, but these are less in terms of frequency than autosomal disorders. Now there are Y-linked disorders, and these are of course rare, as well as mitochondrial disorders. Now these monogenic disorders are important really because 9% of pediatric deaths are due to Mendelian disorders. 3% of them of death are related to chromosomal disorders, and 38% are due to genetic disorders. Now, about 2% of any population has a monogenic disorder. Most of them are dominant, little of them are recessive disorders, and less are X-linked disorders. Now, so Mendelian genetic disorders, usually they symptoms show up at birth. About 1/4 of them are shown at birth, and most of them are shown in terms of symptoms. They are shown right at the age of 10, that is before puberty. Now, we exemplify and illustrate any phenotype, whether it is a disorder or a phenotype, as a pedigree. Okay, so this it shows the family history. So that's how it looks like. Okay, so basically based on this legend right here, square is male, circle is female. If one of them is filled, then it means that the person is affected or carries the phenotype for this disorder. If it is half filled, it means that this person is heterozygous or a carrier. Now, this line means that these two individuals are married, okay, and they have these children right here, okay. So you can see that this daughter is affected, this daughter is not affected, this male is affected, this daughter is affected, this male or son is not affected. Now this means here that this son is married to a female, and they have these two little daughters, and so on. Okay, so if a person's sex is not specified, then it is illustrated as a diamond. Sometimes a number of children of a certain gender is illustrated as shown in here. Okay, now this means that this person is an obligate carrier, and this person does not manifest a disease. If you have an arrow as shown in here, it means that this person is the proband. meaning that this is the person with symptoms, or it's a person affected with a disease that is reporting to a genetic counselor or to a physician that he's having this disease, and he wants to know the inheritance pattern of this disease. Okay, so it's the person with the genetic disorder that goes to the medical community. Okay, if it is filled. Okay, if it is not filled, then it means that this person is not affected. Now this if the symbol is crossed, like in here, it means that this person is dead. This means that that here you either have an abortion or death right after birth. Okay, so this line indicates that these two individuals are married. If the line is crossed, it means that they are divorced. If you have double line, it means that these two people are related to each other. So they can be cousins, second cousins, or whatever. So this is consanguinity. If you have this line right here, it means that these two individuals are monozygotic twins. And without having a line, it means that these individuals are dizygotic twins. Okay, and so on. So I think that this these symbols are important for you to learn. So there are more symbols than that, but these are the basic ones. Okay, so genetic disorders, monogenic disorders, or whatever phenotype can be classified or categorized into different inheritance, into different classes according to their inheritance pattern. So we have autosomal recessive inheritance, we have autosomal dominant inheritance, we have sex-linked disorders, which can be X-linked or Y-linked, and we have mitochondrial inheritance as well. Now, there are some exceptions always, and we'll be talking about these at the end of this lecture. Now, so let's start with the autosomal recessive inheritance. So basically, this phenotype or disorder is caused by a mutated gene, and this gene is present on both chromosomes. Okay, so if one of the chromosomes is mutated, it means that the person is a carrier and there are no symptoms that are shown. Now, the majority of disorders are due to metabolic enzyme deficiencies or autosomal recessive, because these enzymes are important. Okay, so meaning that if a fetus has both alleles being mutated, this fetus would not survive. Okay, so that's why most of them are recessive disorders. Now, so there are certain criteria that indicate that a disorder or a phenotype is autosomal recessive disorders. One of them is that parents of affected people are usually asymptomatic carriers. So these two individuals, for example, this female is a carrier. Okay, so you see that the circle is half filled, and this male is normal. Now they have these children. Now notice that all of them are asymptomatic, and then all of a sudden you have in a family two people who are married to each other, and both of them are carriers. They have affected children. Okay, so basically what you see in here in this pedigree is clustering of affected people in a single family. So you don't see the disease shown in any of the people in this pedigree except this family right here. So the risk for each sibling or carrier to show a phenotype is 25% following Mendelian rules. And something important as well is that males and females are equally affected. Okay. Now, one of the major reasons why recessive disorders are shown is consanguinity. that is marriage among relatives. So you have this marriage of cousins, second cousins, and so on. That increases the risk of being affected with a recessive disorder. Now you can see in here how the distribution of consanguinity around the world, and notice how it's very common in the Islamic world, okay, including the Arab region, like Saudi Arabia, even Jordan as well, and Iraq, Sudan, and so on. So in Jordan, the rate of consanguinity is about 40%, which is really high, and that shows again the disease. Now, one example is Phenylketonuria, okay, which is a metabolic disease. Now, it's not fatal, but it can really affect the quality of life of people. Now, Phenylketonuria, I have to say that it has been, it is widespread in one of the villages in Jordan. That's where you have a lot of consanguinity. Many affected people do live in this village. Okay. Now, what consanguinity does, by the way, is that, remember how I said that recessive disorders are shown as clusters within a family? What consanguinity does is that you would have the disease appearing in every generation. Okay, so it's almost like a dominant disorder, as we will talk about in a few seconds. And that's an important point, by the way. Now, an example of an autosomal recessive disorder is phenylketonuria, which is related to mutation in the gene encoding phenylalanine hydroxylase. So there's a defect in the conversion of phenylalanine to tyrosine. That results in phenylalanine being shuttled into another pathway, being converted to phenylpyruvic acid. And this phenylpyruvic acid would accumulate in the central nervous system, leading to mental retardation. There are other examples that are common, especially here in the Middle East, in Jordan as well, like cystic fibrosis, thalassemia. Thalassemia is really common in the Mediterranean region, and Jordan is one of them. Sickle cell anemia, a lot of enzymatic enzymes in metabolic pathways, and so on. Now, one exception, by the way, and you will learn this in the third year, inshallah, is acute intermittent porphyria, which is related to the defect in the biosynthesis of heme, and it follows a dominant pattern, inheritance pattern. Now, one phenomenon, by the way, is known as locus heterogeneity. Remember, we defined locus in the first lecture, and we said locus is the position of a gene on a chromosome. Okay, so what locus heterogeneity means is that you have many identical clinical syndromes can result from mutations at different genes. Okay, so for example, these are some examples right here. Hemophilia mutations. Hemophilia can be caused by mutations in factor 8 or factor 9, and factor 9 is located on the gene for factor 9 is located on the X chromosome, as well as deafness, by the way. Okay, so but what's important is that if two individuals with deafness, for example, if they get married, notice that their children would be normal, even though that both of them are affected, and you would, and deafness follows a recessive, an autosomal recessive inheritance pattern. This is because deafness in these two individuals is caused by mutations in two different genes. So each one of their their sons and daughters, each one of them would have a good gene, a normal gene, and a mutated gene, and the normal gene would dominate over the recessive mutation, or over the recessive mutation, since we're talking about two different genes. Okay. Now there's another reason for the appearance of a certain recessive disorder, and that is known as compound heterozygote, which means that two individuals, each one of them would have two individuals, parents, each one of them, both of them are normal, and each one of them have a mutation in the same gene, but the mutation is different. Okay, so they have a good gene and a bad gene, or allele, a good allele and a bad allele, and the good allele would dominate over the bad allele. Okay, but their children, or some of their children, might be affected because they would inherit the mutated alleles from their parents. Okay, so they do not have a good copy of a gene. Okay, so most autosomal recessive disorders result from being compound heterozygosity. Okay, except if there are consanguineous marriage. Now, an example is a condition known as Smith-Lemli-Opitz syndrome, which is caused by mutations in the enzyme DHCR7, which is responsible for the biosynthesis of cholesterol. Okay. So just, you don't have to memorize the condition, by the way. Just learn about the concept of what compound heterozygosity is. Now, pseudodominance is, as I said, it's exactly what I said before about the effect, the role of consanguinity. That is, when you see a certain condition appearing in every generation as if it is a dominant disorder, okay, this is called pseudodominance. Okay, and consanguinity again is a cause for the appearance of pseudodominance of a certain recessive disorder. So what is autosomal dominant inheritance then? Basically, all you need, a person would need just one defective gene, or allele, I'm sorry, one defective allele to show the disease, to be affected by a certain condition. And this condition appears in every generation, okay? And males and females are equally affected, or equally at risk. And there are different examples for dominant disorders, genetically dominant disorders, like pseudochondroplasia, Huntington's disease, neurofibromatosis, Wilson disease, and galactosemia. Again, you don't have to really memorize these conditions unless we talk about a condition in more details later on. Now, one of the phenomena that appears in dominant disorders is something known as Pleiotropy. This means that there's a mutation in one gene, but it affects multiple or variable phenotypic traits in an organism. We already talked about one such disorder related to mutations in the lamin A gene. Now, it can cause multiple disorders. So an individual might have the Emery-Dreifuss muscular dystrophy, or the Charcot-Marie tooth disease, or the progeria syndrome, and so on. So depending on where the mutation is located. Okay, so here what you have here is that you have an affected gene, but it can cause different disorders. Now, why this variability exists, no one knows so far for a lot of diseases. Not only that, but there's another phenomenon that is linked to dominant disorders, which is penetrance. So penetrance means that whether a phenotype is present or absent. Okay, so it's white or black, 1 or 0. Okay, so if the phenotype is absent, we say that it is non-penetrant, whatever phenotype is. If we said that if the phenotype is apparent, then we say that it is penetrant. Now, sometimes in dominant disorders, you would get incomplete or reduced penetrance. So there's variability. So it's not 0 or 1, okay, affected or not affected. You can have 0.1, 0.2, according to severity, that is. So you can have severe cases, or you can have mild cases. Now, so you have variable intensity of a phenotype. And this could be caused by variable expressivity, meaning that the expression, the appearance of a phenotype is there or not, mild or severe. So some individuals might have dominant mutations of a gene, yet they are normal. That's penetrance. So why is it that you have incomplete penetrance? Well, it depends on the extent of expression of the mutated gene. If it is highly expressed, then the individual would be severely affected. If it is expressed at low levels, it means that the person would not be affected very much. In addition, it depends on the mutation as well. Some mutations can cause defect in the function of the protein produced from that gene. Okay, some mutations can be mild. In addition, you can have co-modifiers. That is, you can have the presence of modifier genes, by the way, meaning that it not only depends on this particular gene, it can also depend on the expression of other genes as well. So if they are normal, they may compensate for the loss or the mutation of that particular gene. Environmental factors can improve or aggravate the disease process as well. Okay, so even though a person might have a dominant disorder, the severity of the disease can be modulated by environmental factors, or like, for example, lifestyle, eating habits, playing sports, exercising, playing sports or not. Okay, so all of these things can affect the severity of a condition. So an example of a dominant disorder is Huntington disease. Now, Huntington basically, it's the gene has a CAG repeat at the 5' end of the at the 5' end of the messenger RNA or the gene. Okay, so it's a nuclear protein that binds to a number of transcription factors. So what happens is that these affected individuals would have large number of repeats affecting the structure of the protein and the function of that protein. Now, normally, individuals would have about up to 26 of these CAG repeats. Okay? Now, notice that affected individuals having a severe disease would have more than 40 of these repeats. But a person might have reduced penetrance if they have less number of repeats. So higher than normal, but lower than when a person is fully affected by that disease. Okay. Now there is something in between known as it's like a mutable normal allele, or let's call it anticipation, and we will talk about anticipation at the end. So with this anticipation or mutable normal allele, the protein is functioning, but the gene is susceptible to become mutated. The gene with another cell division, the repeats in that gene might increase as a result of an error in DNA replication and replication of this gene. In addition, we have the modifier proteins as well. Okay, so not only that, it depends on the Huntington protein. it also depends on the presence of other proteins that might interact with the Huntington protein. Now, there's a phenomenon known as allelic heterogeneity as well. So with allelic heterogeneity, what you have in here is different mutations in the same gene, but they can cause variable phenotypic manifestation. We already talked about one such disorder, which is the osteogenesis imperfecta, okay, in the cell biology part, when we talked about collagen protein. And so you can have different mutations in the collagen 1A2 gene that encodes the collagen protein, but depending on the mutation, you can have different types. You can have type 2, type 3, type 4, and so on. So there are different types of this condition, and these types can have variable severity. In addition, mutations can cause Ehlers-Danlos syndrome as well. Okay. So, by the way, about 2-3 weeks ago, there was on the news a little girl. Her name is Isra. Physicians didn't know what was wrong with her, and she had to keep on reading and reading in order to know what was wrong with her. And she was able to diagnose herself, and apparently she had Ehlers-Danlos syndrome. Okay, and this girl looks normal, but she's very flexible. She can extend her legs and arms in an abnormal way. And she found herself playing taekwondo really well because she can kick really high because of the flexibility that she has. Now, there are other examples of allelic heterogeneity, that is different mutations in the same gene causing variable phenotypic manifestations. One of them is the fibroblast growth factor receptor 2, which when mutated, it can cause different types of syndromes. Okay, again, you don't have to memorize these, by the way. You might want to memorize the fibroblast growth factor receptor 2 related to allelic heterogeneity, but not necessarily the syndromes themselves. And there's another gene of collagen protein that can cause also different types of disorders with different names and different phenotypes. Now, there's a phenomenon related to dominant disorders or dominant phenotypes, and that is co-dominance, meaning that both alleles are different, but both of them are shown, both of them are apparent. Okay, so a great example is blood typing. So a person can have, for example, we already know that might have the allele for the blood type A. Now, if the person is AA, the person would have a blood type of A. If a person has BB, then this person would have a blood type of B. Now, notice if a person carries two alleles, one of them encodes the A blood type and the other encodes the B blood type, both of them would be apparent. So both of them are really dominant. Okay? And even if they exist by themselves, like A and let's say something else like the O, that is, okay? It means that this person would have a genotype of AO and A dominates. Same thing with B. Okay, so this is known as co-dominance. Okay, so let's get into sex-linked inheritance. Now, with sex-linked inheritance, it means that the defective gene is present on the X chromosome. Okay. Now I have to talk about a phenomenon that we already talked about in the molecular biology lectures, and that is X chromosome inactivation. That is, in females during development, you would have in every single cell one of the X chromosomes being inactivated. Now, we already talked about the mechanism of inactivation. We said that it is related to a long non-coding RNA known as ZIST, XIST, which coats the X chromosome that carries the gene, and it results in acetylation and methylation of histones and the DNA itself. Okay, resulting in shrinking of the X chromosome and inactivation of that X chromosome. So it looks like a dark particle inside the nucleus, and it's called the Barr body. So the inactive X chromosome becomes what is known as a Barr body. So pay attention to this phenomenon, because if you have a mutated gene on the X chromosome, you have to ask yourself, is it located on the active X chromosome or the inactive X chromosome? Okay. Now, why is it that we have X chromosome inactivation? Well, this is known as the term is related to what is known as dosage compensation, meaning that females have 2 X chromosomes, so and males have 1 X chromosome. So one might expect that females would express double the amount of proteins from the X chromosomes versus males, but this is not the case. Having 1 X chromosome inactivated would enable female cells to express the same amount of proteins as male cells. So X-linked disorders, genes are located on the X chromosome. Since males have 1 X chromosome and we call them homozygous, the presence of an abnormal gene on the X chromosome would be dominant, by the way, even if it is recessive. Okay. Now females have 2 X chromosomes, so they are heterozygous. Okay. Now, since we have this phenomenon of X chromosome inactivation, that would affect the inheritance pattern, or let's say the showing of symptoms on the X chromosome. Okay, so again, pay attention to that, and we'll talk about it in a few seconds as well. Okay, so the idea here is that, let's say that you have a recessive gene. Okay, and this recessive gene is located on the active X chromosome. Okay, this means that the good gene would be inactivated, and the symptoms would appear on that person, on that female. Okay, again, it depends also on the organ that is affected. Is this gene important for that organ or not? Because the way X chromosome inactivation takes place is that it's random. So you can have liver, for example, having 50% of cells having this X chromosome inactive, and the other 50% would have the other X chromosome inactive. Now this ratio can be different, so it can be 70 to 30%. Okay, so depending on the number of cells that are affected, that have the mutated gene being on the active X chromosome, would affect whether symptoms would show or not on any person. So these are the criteria of X-linked recessive disease inheritance. So it affects males only, so this is really important. So that's because if a female is a carrier, there's a 50% chance that males would be affected, because they will get 1 good chromosome or 1 bad chromosome. Okay. Now, affected males are born to unaffected parents, so the mother is normally asymptomatic. She's a carrier, of course. Females may be affected if the father is affected, right? Because she's going to take just this 1 bad X chromosome from the father. Okay. Now, a woman, I have to fix this. A woman that carries X-linked recessive disease usually is physically normal, but symptoms can appear depending on on X chromosome inactivation. Again, there's a 50% chance that a carrier female will have an affected son and a carrier daughter. Okay, and sons receiving the Y chromosome are free of the affected males have normal offspring, normal male offspring, right? Because they will take only the Y chromosome from their father. But daughters would be obligate carriers. So this right here, this symbol right here, it means that she is an obligate carrier because she would get this bad X chromosome from her father. Okay, same thing. She's a carrier as well. Now, what are the cases where you have complete expression of an X-linked recessive disease? When the allele frequency for this disorder is high in this population, or let's say in this family. Again, consanguinity plays a major role in here. A female hemizygous for an X-linked trait, such as Turner syndrome, would also have the disease as well, since this female would have only one X chromosome, and this one X chromosome would carry the mutated gene. Now, X-linked recessive disease can appear in females with deletion of the same allele of the normal X chromosome, that is the active X chromosome. Now, you can have faulty X inactivation, where you have both alleles being inactivated. Okay, this is a rare phenomenon. Or you can have skewed X inactivation, meaning that there is a high probability, instead of having 50-50% of inactivating this X chromosome or the other X chromosome, you might have skewed X inactivation, meaning that you have 80-20. Okay, so being the with 80% of the faulty X chromosome being activated in the body of that individual. You can also have what is known as X autosomal translocation that causes Duchenne. An example is Duchenne muscular dystrophy as well. Okay, so in this case, you would have a replacement of the good gene by a bad one on the active X chromosome. Now, what are the features of an X-linked dominant disease inheritance? The child of an affected heterozygous female, regardless of its sex, would have a 50% chance of being affected. Okay, so you can see this pedigree right here. This is an affected female, so there's a 50% chance that her offspring, sons and daughters, would be affected. Okay, for an affected male or daughters, but none of the sons would be affected, right? So also this right here, again in this pedigree, this is an affected male, so all daughters would be affected. Male to male transmission never happens, of course. So again, this illustrates significance of X inactivation. That is, a recessive gene in a carrier female is occasionally expressed due to inactivation of significant number of the X chromosomes containing their normal gene. A dominant trait, by the way, in a carrier female, this female can escape expression of that disease. Why? Because the faulty gene might be located on the inactive X chromosome. So, an example of an X-linked disorder is hemophilia. Okay, so mutations also affecting coagulation cascade that result in hemophilia follow locus heterogeneity, basically, which means that you have hemophilia, but mutations can be present on different chromosomes. So you can have inactivation of or mutations in factor 9 or factor 11, factor 9, or factor 8. Okay, so the most common is known as hemophilia A, where you have defect in factor 8. Okay, so and you have also, you can have 620 different mutations in the gene in the gene of factor 8 that causes hemophilia A, and this is an example of allelic heterogeneity, right? So you can have different mutations of a single gene causing the same condition but variable symptoms. Now, hemophilia B results from mutations in factor 9. So coagulation basically is the final formation of a fibrin as a result of activation of all of these enzymes, assisted by a number of factors and cofactors. Okay, so mutations in any of these enzymes would cause hemophilia, which is illustrated by excessive bleeding because of the inability of an individual to form fibrin to stop bleeding and to cause blood clotting. So Duchenne muscular dystrophy is also an example of an X-linked disorder. Usually it affects young boys, although they would look normal at birth, but but as they grow, the symptoms would be exaggerated, and it can be fatal when they reach their 20s or so. Now, Y-linked disorders are related to mutations on the Y chromosome. There aren't that many genes on the Y chromosome, but they are important for the maleness of individuals. Now, as you can see from this pedigree, the pattern is you have dominant transmission of this disorder from males to other males. So all males will be affected. Okay, and there's no female to male or female to female transmission, of course. How about mitochondrial inheritance? Now, mitochondrial DNA is maternally transmitted. Okay, so it follows this transmission from females to offspring, daughters and sons. Now, most mitochondrial proteins are encoded by the nuclear DNA, not the mitochondrial DNA. So most mitochondrial disorders follow the inheritance pattern that we just talked about. That is autosomal recessive, autosomal dominant, X-linked, and so on. Okay, but you can have certain diseases that are related to mutations in the mitochondrial DNA. The organs that are most susceptible, by the way, due to mitochondrial mutations are the central nervous system, skeletal muscle, and heart. Okay. Now, affected mothers transmit diseases to their daughters and sons equally, and only daughters would transmit the disease to her offsprings or to their offsprings. Now, symptoms vary according to the to certain conditions, such as so they vary according to severity, the age of onset, the affected tissue, and so on. And it is related to the proportion of mitochondria that carry the mutation, by the way. So, for example, in a young individual specifically, they would have mitochondria having identical mitochondrial DNA. But you can have people with what is known as heteroplasmy, meaning that mitochondria in a single cell would have different genetic component and different genetic component in terms of mutations and sequence and so on. And it increases with age, by the way. So if there are very low number of affected mitochondria, then the person would show no disease. Okay, but if you have all or large number of mitochondria carrying the mutated DNA or mitochondrial genome, then the person would have a severe disease. If it is like 50-50, let's say, the person would have a mild disease. So there's variability in the severity of the disease. Now look at the pedigree and notice the inheritance pattern. So it goes from females to, again, to sons and daughters, and only daughters would transmit the disease to their offspring, by the way. And we already talked about this a three-parent baby in the molecular biology part of this course as a solution of this disease. Now let's go over other modes of inheritance. One of them we talked about before, which is anticipation, which means that the genetic condition would tend to become more severe and appearing at an earlier age as the disorder is passed from one generation to the next. So there is sort of like a qabiliyah, so there is sort of like a susceptibility of a person being affected with a certain condition with ongoing generations, successive generations. Okay, and usually this anticipation occurs when disorders are caused by trinucleotide repeat expansion. And examples include Fragile X syndrome and the Huntington disease. So the Huntington disease is caused by a CAG repeat in the Huntington gene. So the idea here is that with every round of DNA replication, the number of repeats would increase, resulting in increasing severity of the disease. The age of onset becomes lower, and so on. An example is Fragile X mental retardation syndrome, or the Fragile X syndrome right here. Usually, so the most common form, or FMR is the most common form of inherited mental retardation. It results from the end of an X chromosome being fragile, so it gets broken off the chromosome. It's an unstable part of the chromosome, and it results from mutations in a repeat of CGG in the coding sequence of the gene FMR. Okay, so the inheritance of FMR is unusual, and about 20% of males with Fragile X syndrome are phenotypically normal. Okay, but they can transmit the affected chromosome to their daughters, and they would appear normal as well. Now, these males are known as normally transmitting males. So they are normal, but they transmit the faulty chromosome to their daughters. They look normal, and they can transmit the faulty chromosome to her offspring. Now, this phenomenon, by the way, is known as permutations. Okay, so permutations is like, it means alteration, change. So what happens here is in a normal individual, a person would have, the number of repeats would be about 6 to 54 repeats. Okay, now in normally transmitting males and their daughters, the number of repeats increases from 50 to about 200. Okay, now so this increase, by the way, is known as permutation. So it causes the gene to become unstable, but affected individuals would have large number of repeats, about 200 to 1300 repeats. So again, basically a normal individual would have about 60 to 50 copies. Okay, so just I'm not gonna ask you about is it 50 or 56, by the way. So you can see there is a variability among textbooks and papers and figures as well. So an individual having up to 50 copies would be normal. Now 55 to 200 copies, this individual right here would carry what is known as a permutation. So the allele is unstable. So when this chromosome is transmitted to the offspring, the number of repeats would increase to more than 200 copies, and this is a fully mutated gene. So this repeat exists in the 5' untranslated region of the FMR gene. And this gene is located at the end of the X chromosome. And the increasing number of repeats causes the X chromosome to become unstable. So it's like it gets broken off from the X chromosome, and that's why they call it the fragile X syndrome. So, like other diseases as well, penetrance is variable. Remember, penetrance is having a phenotype or not having a phenotype. But here, the penetrance can be low or it can be incomplete as well, okay? So an asymptomatic son, notice here. So here you have a 4th generation. This is a female that is not affected, but she has an increasing number of repeats. She transmits the gene to her offspring, and notice that this boy is affected. This boy has a permutation. Okay, now this is a normally transmitting male right here, because he transmits the chromosome to his daughter, and his daughter is normal because again she has 2 X chromosomes, right? So she's a carrier. But notice what happens here is that all males would be affected by this disorder. Now sometimes you can have the pedigree drawn like this, showing the number of repeats. So again, right here, you have that's a normal female, right? Now notice here that this male has a permutation. This female is normal as well. Now this is a normally transmitting male. Okay, he transmits the faulty chromosome to his daughters. Now this daughter would transmit the disorder to her offspring, and they would be affected. Same thing here. Again, so these are features and rules that you can follow in terms of in order to determine if a certain disorder is follows the pattern of autosomal dominant or recessive or X-linked or Y-linked and so on. Let's talk about mosaicism. We talked about it before, and we said that basically it's a person having the same genotype in all cells, but some cells have a mutation that does not exist in other organs or other tissues. And this happens during development. So let's say here you have a zygote, okay? So you have multiple divisions of the cell, and all of a sudden you have a mutation in this cell right here. This results in the formation of several cell lines, and all of them would carry the mutation. Now, so you might have an organ that is affected, like the brain, liver, kidney, whatever, depending on to what organ or tissue does this cell differentiate into. Okay? So mosaicism, by the way, can be somatic or they can be germline. So you can have the cell that is affected, a somatic cell. So you would have this individual having an affected organ, for example, or organs. It depends on, again, to what tissues or cells does the affected cell differentiate. Or it can be in germline. That is, you see that the mutation occurs in germline cells, but not in somatic cells of that individual. So you would notice that all children would be affected, right? So this is a normal male right here, okay? But you have mutation in some of the germline cells, and you would notice that one of their children would be affected by a disorder, even though the parent, the father, is really normal. Okay? But it turns out that the mutation exists in the germline cell. So this is mosaicism. Now another phenomenon is known as uniparental disomy, meaning that you have 2 chromosomes coming from one uniparent. Okay, so this is uniparental disomy, and this happens again during differentiation, during meiosis. What happens here is that you have a normal segregation of chromosomes, right? But then all of a sudden you can have non-disjunction right here, okay, resulting in a cell carrying a two chromosomes, having two chromosomes, being disomic. Okay. Now, if you have fertilization happening, you would have a zygote with three of the same chromosome. Now, sometimes what cells do is that they degrade this one chromosome, or they kick it out of the cell. Usually, they degrade them. So this germline cell would have, this zygote, I'm sorry, would have two chromosomes coming from one parent, okay, but not the other. Now, you can have it, by the way, this phenomenon, the non-disjunction, in meiosis 2, or you can have it in meiosis 1. But the idea is the same, basically. That is, you would have a zygote with three chromosomes, and one of these chromosomes would be removed. Okay, so it appears that in one condition, you have a child with cystic fibrosis, even though the parents are not affected. But it turns out that both chromosomes in this little child came from one parent. So this is uniparental disomy. Now there's another phenomenon known as genetic imprinting, or in other words, the parent of origin. So the idea here is that there are about 80 genes that must be expressed from one of the parents specifically. Okay? There are 80 genes that must be expressed by one of the parents. So this particular gene, it's the maternal gene that must be expressed, or the maternal allele that must be expressed. And there are other genes that the paternal allele must be expressed, not the maternal allele. Okay? So this is a result of genetic imprinting. So what happens is that if the maternal allele must be expressed, the paternal allele would be methylated, right? Epigenetics. So it becomes inactive, and vice versa. The opposite is true. Now there are two conditions known as Prader-Willi and Angelman syndromes, and both of them are caused by deletion of a distinct region on chromosome 15. What happens, it has been shown that even though the same region is deleted, two different syndromes would appear. Now, the Prader-Willi gene with deletion of the paternal gene, or maternal uniparental disomy, meaning that what happens here is that if the paternal gene is deleted, it would cause a Prader-Willi gene. If the maternal gene is the one that is defective or deleted, this would cause Angelman syndrome. Now, another reason for showing either Prader or having Prader-Willi gene or the Angelman syndrome is if you have uniparental disomy. That is, you have both chromosomes, chromosomes 15 coming from the mother. Okay, not father, mother, both of them coming from the mother. Then this individual would have the Prader-Willi syndrome. Now, same thing with the Angelman syndrome. If there is paternal uniparental disomy, the person, meaning that there is no maternal gene or maternal allele, then this person would have Angelman syndrome. So, what is the mechanism behind having either one of these syndromes? See, it's all related to that region, okay, on chromosome 15. There is a regulatory sequence. It's known as the imprinting control region. Now, this region controls expression of genes. The genes that are regulated are known as UBE, which has to do with it's a ubiquitin labeling protein. and three other genes located on the other side. And there is a gene right here that encodes a non-coding RNA. So the idea here is that in the paternal allele, you have this ICR region. Okay, what it does is that it results in the unmethylation of these genes, so they become active from the paternal chromosome, and activation of this gene right here, which encodes this non-coding RNA that inactivates the UBE gene. Now, this gene must be activated from the maternal chromosome. These genes must be activated from the paternal chromosome. Okay. Now, what happens on the maternal side, the maternal chromosome, is that you have within the same region. What happens is that you have inactivation of production of the non-coding RNA, resulting in activation of this gene, and at the same time, this region is inhibited, resulting in methylation of these genes, resulting in their suppression. So these genes are suppressed, they're not activated, and this gene would be activated from the maternal chromosome. Now, so these two conditions are caused mainly by deletion of the region, either the paternal chromosome or the maternal chromosome. So most of them are caused by deletion of that region, of that chromosome or the other. Now, uniparental disomy can also be a cause, or you can have a mutation in that particular gene from the maternal side, or you can have imprinting defects as well on both chromosomes, but mainly it's deletion.